tailored to all wishes, let's say. So there'll be a few things which are a bit more general. So for the specialists in the audience, that may sound a bit familiar. And hopefully there's also some novelty things for those ones which maybe might be a bit outside the comfort zone of some of the less experienced people. But I basically just want to start a little bit first with CBS. I'm not sure if all of you are very familiar with CBS. This is the building down here, sitting next to a neighboring institute. So CBS probably has about 70, 80 people working there, so it's a fairly small institute, but it is probably still the leading international fungal collection, at least the public fungal collection. So we currently have about 75,000 to 80,000 strains in our collection. And the nice thing about it is that within, here still says within 3 years, I keep forgetting to change that slide. It should be now within 1.5 years, they will all have a DNA barcode. So that actually means that all of them have been identified at the molecular level, which is something which those of you who have been around a bit longer know is quite crucial, because morphological taxonomy can be quite misleading. And what we basically want to get at CBS is that ultimately we want to have a gold standard effectively for identification, where you can maybe not always be certain about the species, because that's not always possible with a single barcode, but at least you know you're working with the right genus. So,
and who supports it financially, and what is it all available?
Yeah, so basically, that's a good question. So the barcoding has been supported by various sources actually, and the DNA barcodes as such, as a database, will not become publicly available, because that of course would be giving away a lot to our competitors, let's say. What will probably become available, but that is still in development, is an option that you can have your own sequence mirrored against the database, and that will give you the most likely identification. So we are currently developing a service that you can use this database to identify your own species if you have your own sequences, but we're not very keen on just giving out 75,000 barcodes to the rest of the world, because then of course our competitors in China and US and other places could just download those databases and provide the same service to other ones. And to some extent, as you said, for financing, to some extent, these things are funded by the sales of our strains. So we would like to keep that.
So all 75,000 are for sale?
All 75,000 strains will be barcoded. Yeah. And in addition to that, all the 3,000 to 5,000 new ones which come in every year. So we still grow with about 3,000 to 5,000 strains per year. And so yeah, it's quite a serious job. There's about 20 people at CBS involved just in maintaining the collection. Now then, in addition to the collection, we have 5 research groups. We have applied mycology from Rob Sampson, we have phytopathology from Pedro Kraus, medical fungi from Steven de Hoog, and yeasts from Teun Boekhout, and then my group, which is fungal physiology. So my group is a little bit, let's say, the odd group out at CBS, so most of the groups are very much into biodiversity, taxonomy, and species identification and description. We are not. When I was hired specifically to do something with the collection rather than to add to the collection. So we don't do very much in the sense of species description. We mostly try to use fungal diversity to understand our favorite topic. Now just a couple of pictures so you have some idea of what is involved in maintaining such a collection. So here you see all kinds of climate rooms on the right top. Here you see a large part of our collection in the bottom, which is liquid nitrogen vessels. So that we have, I think, currently 18 of those. And they have 12 towers with 12 boxes each with about 100 samples per box, so you can do the math. In addition to that, we have lyophilized collections, so it's quite a serious undertaking. Anyway, what I was going to talk about today, which is my favorite subject, is how fungi use natural substrates. So this is a topic I've been working on more or less since my PhD. I had a few little ventures into other directions, but still managed to hobby around on this topic, so was lucky to sort of have a no break in publishing in this. And for many years, this wasn't very popular, especially if you look at plant biomass. But of course, more recently, with all the interest in biofuels and biochemicals, this has become a very hot topic, and a lot of groups have joined this field, so it's quite a strong field. But just to give you a bit of background, so if you look at fungi from a more, let's say, academic, natural point of view, you know that most fungi can grow on many different substrates. But if you look across the fungal tree of life, plant biomass is one of the most dominant carbon sources for fungi. And this is, of course, mostly due to the fact that it's the most abundant carbon source available on Earth. But what's very important then is that, of course, the lifestyle of these fungi are quite important for their strategy on how to deal with biomass. And you can mostly divide the lifestyles into three main topics. So we have the saprobic fungi, which use the dead biomass. And in many cases, biomass has already undergone some kind of, let's say, pre-treatment. It could be a branch which fell off a tree and shattered partly, so it has more openings, more accessible. It can be partly degraded by bacteria or other microorganisms. So this dead plant material is not necessarily intact. Then, of course, we have the pathogens. They do deal with intact plants, and they, of course, not only have to degrade the plant biomass, they also have to overcome the defense mechanisms of these plants. So they need quite a different strategy for that. And then, of course, we have the symbionts. And the symbionts, of course, there are many different types of them, like for instance the mycorrhizae. They have to be, let's say, nurturing their host. So they still need to make some kind of damage to the host to be able to form the interaction. But then at the same point, they need to make sure that this damage is not, let's say, life-threatening for the host, because the host is very important for their lifestyle. So these different lifestyles require very different strategies. And one of the things we're doing in my group is trying to understand from both genomic perspective, but also from a more physiological perspective, to try and figure out what are now the criteria which determines these lifestyles, and can we ultimately maybe cluster fungi, even fungi which are poorly understood, in their ability to either be sapro, pathogen, or symbiont. Now, just a little bit on plant biomass for those of you who are a little bit less involved. So when we look at plant biomass, we mainly look at polysaccharides, like things like cellulose, hemicellulose, and pectin, but also storage carbon sources like starch and inulin and gums, and of course also lignin and protein. And in addition to that, of course, this composition of plant biomass varies very much from species to species, from tissue to tissue, and from season to season. So for fungi to be able to degrade this, they need to be quite flexible. Now, just a little picture which I always like to show, because I think it's still one of the nicest graphs of a plant cell wall. So here you see the intricate structure. So here you see the microfibrils from cellulose, which are forming the skeleton, and they're intertwined by pectin and hemicellulose chains, which basically give it the nice interactive structure that allows the plant to grow upright, but also allows the plant to prevent attack by many microorganisms. Now, you will hear me talk a little bit about models sometimes. You will not hear me talk about model fungi, because I personally don't believe there's a model fungus for plant biomass degradation. And this is because what we found is that all fungi are actually quite different when it comes to this. But we still use models, but we use more, let's say, strategic models. And I always like to point out that in my opinion, every model is wrong. And because a model is based on what you know at this point in time, and we never know everything. But the good thing about a model is that it helps you actually find those cavities in your knowledge. So this is our basic model for the use of plant polysaccharides as carbon sourced by fungi. So here we see a very simplified fungal cell, which has a nucleus. It probably has some random transporters always in its cell wall or cell membrane. And at a certain point in time, it finds itself in the presence of a monomeric compound, which it may have some interest in. So the first thing that happens, it has to realize it's there. So it has to be sensed. Sensing in fungi, especially of sugars, is very, very poorly understood. There are a couple of examples of that, and they are very non-homologous to each other. There's, of course, a lot of information in Saccharomyces, but I don't think we can really use that as a model, because that's quite a, let's say, adapted species of fungus. So I would say there's two possibilities for sensing. One is, which is the classical way of sensing, which is basically a sensor sitting in the cell wall, sensing the compound while it's still outside, and basically starting up some kind of response to get it inside. I think, though, it's also equally possible that we have these transporters, which are, let's say, broad substrate range transporters, which just take up anything which is in the vicinity of the fungal cell, and then the sensing may actually take place inside the cell rather than outside the cell. Well, however this sensing takes place, this is followed by a signaling pathway, which ultimately results in the activation of a transcription factor, which in the nucleus will bind to the genes it's targeting. Now, in the case of polysaccharide-related utilization, usually then two things will happen. One is that, of course, the metabolic pathways required to convert this compound are activated, because otherwise there's no point for the fungus to actually take up this carbon source. But in nature, as most fungi, of course, are quite aware of, there is a very, very low supply of monomeric carbon source, because as soon as there's a monomeric carbon source around, something will eat it. So if you don't eat it, your neighbor will. So the presence of a small amount of this monomeric compound indicates the presence of a larger amount of a polymeric compound. So at the same point with activating these metabolic pathways, also extracellular enzymes are activated, which basically can degrade this polysaccharide to create more of this monosaccharide. Now, this is a very simplified form, but of course the nice thing about this is that as soon as this polysaccharide runs out, the monosaccharide is no longer formed, and this whole cycle basically gets terminated, and the fungus will either go dormant or will move to some other substrate. Now, this is of course a nice way to, at least look at the overall system, because it allows you to put different aspects of this physiology into relation with each other. So today I'll mostly talk about extracellular enzymes and transcription factors. We also do quite a bit of work on the metabolic pathways, but in light of time I decided to leave that out for today. So when we look at polysaccharide degrading enzymes, there's mainly two questions you can ask. One is that, of course, which enzymes do we need for which polysaccharide? And this is something which became much more clear since we've got genomes. But of course the question is, how do they actually act? Are some enzymes mostly acting by themselves, or is synergy really a crucial factor in this? I just want to give you a few highlights on that, some actually from quite some time ago. So just to give you a bit of a rundown of what you get, so we've got some simple polysaccharides, so like cellulose, starch, and inulin. And in most cases, there's only about 3 main enzymes, although there are some additional enzymes which, let's say, contribute to degrading these polysaccharides. But in most cases, with 3 main enzyme groups, you can get pretty far at degrading these polysaccharides. Then in addition to that, we also have the more complex ones. So we have, for instance, xylane, and immediately in this cartoon you can see the difference in complexity. So still we have this monomeric, let's say, backbone, but then we have all these different side chains, and we need many more different enzymes. So in the case of xylane, we get a much larger set of enzyme activities we need, and of course there may be still quite a few which we are currently unaware of. So depending which polysaccharide is there, you need to decide as a fungus which enzymes you need to make. Now, a lot of the things I've mentioned before, I could have told before we got genomes, and this topic is something that particularly has been looked at for a long time in Aspergillus, and I worked in Aspergillus on this topic before we had genomes, and I think we were a little bit overconfident in those days. We tried to purify every enzyme we could find when we grew up the fungus, and then we tried to clone the genes, and I think before we had genomes, we had about 40 genes cloned involving plant polysaccharide degrading enzymes. And we were pretty convinced that we got more or less everything. Because we could degrade every linkage we could find, we thought, okay, we're there. Then we got a genome. And the first Aspergillus genome which came out, which was of course Aspergillus nigricans, followed quickly by Aspergillus oryzae and fumigatus. I think the first thing it told us is that it really told us we didn't know that much. Because if you look at Aspergillus genomes for these kind of genes, you can easily find between 160 and 250 genes. So clearly, we don't know exactly what's going on. And I think one of the reasons for that is, of course, that fungi in nature grow mostly on solid surfaces. They change what enzymes they produce as they grow because the substrate changes as they start degrading it. Whereas, of course, in the lab, we grow these fungi in shake flasks or in fermenters in fairly homologous or homogeneous conditions. We basically use very simple substrates. So how can we actually assume, looking back now, it's easy to say, that we actually will be able to see all the enzymes in those very simplified forms of fungal growth? So this makes a lot of sense. So we have much larger enzyme systems. But then, of course, if you're looking at things from a, let's say, diversity biotope perspective like we like to do, the question, of course, is can we then use this genome information to understand the biotope of fungi and their ability to use these substrates? So can we actually correlate genome content to physiology? I'm just going to show you a few examples of that, but before that, I just want to introduce this. Most of you are probably familiar with the CASI system, so I won't linger here very long, but for those ones who are not, so CASI is a very useful database for anybody working on plant biomass degradation because it clusters all the enzymes related to this topic in very nice families, which sometimes only have a single enzyme activity and sometimes more than one, so then you need to do a little bit more work. But it really helps you very quickly to sort of get the degradation potential of a certain species if you have a genome. So this I'm using also quite heavily in the rest of the data I will be showing. So let's start very simple. This is a slide I've used before. I've added the Rospera here because of certain interest of people in the room. And so what you can do very easily, you can have a look at the potential of a fungus. So here I compared two substrates, pectin and xylane. And what I've done is that behind here you see the CASI families which contain enzymes only involved in that polysaccharide degradation. So there's also families which contain enzymes for both of them, so I've left those out deliberately because they would sort of confuse the situation. And what you can see then very nicely, if you look here at pectin degradation, Aspergillus niger and botrytis, they're very rich in it. Actually, that yellow in Neurospora should not have come up. Also, Rhizopus oryzae is quite rich. The other three are quite poor in pectinases. In contrast, if you then look at the xylanases, we see a very, very rich content for Podospora and Serena, reasonably good for Phanerochaeta and Neurospora, a little bit less for Aspergillus, and absolutely nothing for Rhizopus oryzae. So this is what the genome tells us. The question is, does this actually then mean something? So we could test that very simply. So we basically just did some growth profiling. You'll see we did Neurospora a little bit different. And what we can then do is have a look what actually comes out. Now, a few things, of course, are interesting in surprises. So we saw that Aspergillus niger was more rich in pectinase than in xylanases. But if we're looking here at xylan and pectin here and its monomeric components, it actually grows quite happily on everything. And this is typical for Aspergillus, I would say. Aspergillus seems to be the typical generalist. It likes more or less everything. It has a broad set of enzymes, maybe a bit more for some than for something else, but it covers more or less all activities. Now, then the light is not perfect, but what you see here in botrytis, we see growth on pectin, but we don't see growth on xylan. We also have some growth on monomers, and the same here for Rhizopus. It's very thin, so probably in the back you can't see it, and I have to just take my word for it. We have growth here, and we have growth there. We don't have growth on xylan. And if we go back to the previous slide, you can see Rhizopus, no xylanases, botrytis, much less than pectinases. So in those cases, we see a very nice correlation. If we then look at the other side of the spectrum, Podospora and Serena, very rich in xylanases, very poor in pectinases, perfectly fits with the growth profile. Then we have Neurospora, a little bit confusing at times, because actually it doesn't like this xylan so much. There's actually other xylans we tried it does like. So it seems to be a bit fussy when it comes to xylan. It doesn't like beechwood xylan. It doesn't mind birchwood xylan. It doesn't like oatspale xylan very much. So I must say I'm not a Neurospora specialist, so I haven't quite figured out why it does that. In most cases, we see quite nice correlation that if it's good on xylan, it's good on all xylans. Neurospora is a little bit more difficult there. It does like the monosaccharides. And on pectin, we had a very strange result. We actually repeated this once more, but again 3 tubes, and again we had 1 tube where it grows and 2 tubes where it doesn't. I can't explain this. Maybe some people can give me some hints over lunch later or something. But anyway, so what we find is that for many of these fungi, we get these kind of correlations. Now, of course, we could say this is 6 fungi. What does it mean? So that's what we thought as well. So in addition actually to these carbon sources, we in total tested 35 carbon sources for all these fungi. And by now we've tested about 400 fungal species on this. And interestingly, what we find is that for about 95% of the fungi, there is a direct correlation between better growth and a more extensive enzyme set. Also interestingly, one of the most typical fungi studied in this field, Trichoderma raceyi, which is the, let's say, model fungus for cellulases, it's very atypical. So Trichoderma raceyi does not produce many different cellulases, it produces a few of them at very high levels. But this is not a strategy which is common among fungi. So what we find is that I would say 95% of the fungi follows the strategy, grow better, have more enzymes. Then there's a couple of other ones, and Neurospora is sitting very much on the Trichoderma side. It's not as extreme as Trichoderma, but it's more focused enzyme set, which apparently is being produced at a good enough level to be able to work. Now, still, we could use this, of course, so we could use these for all these, let's say, 400 fungi to try and see some general patterns. Before that, I want to give you a few little other examples, so that even the species I showed you before, they're quite distant from each other. So what happens if you do a similar comparison, but you look within one genus? So here you see 3 Aspergilli, Nigellans, Niger, and Oryzae, still, let's say, a decent taxonomic distance from each other. And what you can see is that they differ quite dramatically in certain growth. So this is growth on glucose. That's always our internal reference. So we do not compare, let's say, fungus A on xylan to fungus B on xylan. No, we compare the relative growth on xylan to glucose in fungus A to the relative growth on xylan to glucose in fungus B. So this is also what you need to do here. So take into account that Niger is a bit smaller here than Nigellans and Oryzae as well. Still, if we look here at galactomannan, we can see that relatively Oryzae doesn't like galactomannan very much, whereas the other 2 are quite happy. But more dramatically here in the case of inulin, we see that only Niger grows really well on inulin. Nigellans barely grows at all, and Oryzae grows much weaker. Now, inulin is particularly interesting because there's only one Casey family involved in inulin degradation, that's GH32. So it's very easy to compare these fungi with respect to genome content. And if we do that, so basically we look here at inulin at the bottom, what we see is that Niger has 5, Nigellans has 2, and Oryzae has 4. Okay, so nitrogens being bad makes sense. It's much less, let's say half the enzymes which nitrogen or ricei have. But why is there such a big difference between nitrogen or ricei when they only differ in a single enzyme? Now, we haven't tested it. I have to say that we're still on the list for the next undergraduate student to do this. But the nice thing is, if you then start looking into these enzymes a bit more in detail, you look at the specific activities of them, what we find is that the difference between nitrogen and ricei is that nitrogen contains an endoinulinase, whereas ricei only contains exoenzymes. And this makes perfect sense then, because if you have an endoacting enzyme, you can cleave your polysaccharide into oligosaccharides, and your exo can really quickly degrade this into monosaccharides. If you don't have an endoenzyme, the only thing you can do is go from the outside of your polysaccharide and chew your way in, which is a much more tedious process, of course, than making oligosaccharides. So we're still planning to do it. We still haven't, well, I would say it's not high priority enough, clearly, for us to have done it already. But of course, what we want to do is put this endoinulinase in Aspergillus oryzae, and hopefully we will see that the growth in oryzae will be actually similar to the growth in nitrogen. So in some cases, if you look at this genome content in a bit more detail, you can actually explain a phenotypic difference by a single gene. And I think this, of course, makes for some quite interesting stuff also for applications. Now, as I said, we've done probably about 400 species now. We've not got them all uploaded yet. But when we started doing this initially as a hobby, and when we realized that this was going to be quite interesting, we started thinking about how to present this to the scientific world, because there's only so much data you can put in supplemental files in a paper. And if you take into account that we have 35 carbon sources per species, so 400 times 35, you can do the math, this is a little bit too much to add into supplemental data. So what we decided to do is to make a database. So that's here. If you just basically search for fun growth, you pretty much should get it. We initially had the idea of doing fun growth, but that got very strange hits in Google, so I decided not to go for that one. So fun growth works. That gets you to the right place, so don't forget the G. Basically, I can explain to you how the database works, but it's not necessary, because at the starting page there's this button called video instructions. If you just follow that, what you can basically do, you can pick your favorite fungi, you can pick your favorite carbon sources, and it will generate for you on the screen a comparative picture of all the growth on them. So you can basically have a look how one fungus compares to another without having to do all the data. And hopefully my current bioinformatician is working on a more simpler script to actually upload the rest of the data we still have. So hopefully sometime this year we will have about 400 species in there. I think it's about 150 or 180 now. Now what's in there? So basically we decided to do 35 different carbon sources. So we have all the monosaccharides which are commonly found in plant biomass, then some of the oligosaccharides. You might notice this one here, lactose, not exactly related to plant biomass. Unfortunately, there's no commercial beta-linked galactose oligosaccharide which is related to plant biomass. So we basically decided to just take the next best thing. Then we have a bunch of different polysaccharides. We have a number of crude plant biomass substrates in there, and of course a few controls like no carbon source, because some fungi do like agar. Even though we use a very pure agar, there's still some of them quite happy still to grow on that. We included the lignin as a carbon source and protein, because some of the crude plant biomass substrates also contain protein, so we want to exclude that. So this is all in there, and then if you have that, and especially of course if you're sitting in CBS where all that data is sitting on your computer, you can start doing fun things. And one of the things we started doing is to start doing correlation analysis. Because once you get big data sets, this is the best thing. So we have actually quantified growth. This is not yet in the database, because we want to wait until we publish this. So what we did, we took a scale of 0 to 10, where we quantified the growth, and then we basically put this in a cluster analysis over a basic set of 22 fungi. We're now expanding that to include the entire set, but this is on 22 fungi. And the nice things you see then is that you get some really interesting details. So like here on the left-hand side are most of our plant biomass substrates with citrus pulp on the end sitting right next to the pectins. Well, citrus pulp is very rich in pectin, so that makes a lot of sense. Interestingly though, cottonseed pulp, which was also one of our crude plant biomass, is sitting all the way over here, and it's sitting next to lignin. Now this initially puzzled us, because we were thinking, why the hell does that one split off so much? So then we had the composition analysis done on all these substrates, also including the lignin content. And what we found is that all these substrates over here are between 0 and 3% lignin, whereas cottonseed pulp is about 25% lignin. So what we're getting here is that one component of the plant biomass, in this case lignin, is so dominant in the effect it has on fungi, either by the ability for the fungus to degrade it, or even just the ability to tolerate it, that this is basically dominating the growth on this crude plant biomass. So that's actually, we were quite happy with it to see that, because it basically means that we can, by these type of correlation analysis, we can see which components of the plant biomass are particularly important for fungal growth, and maybe even for the ability of fungal enzymes to act. Now, we also saw a few other things which are interesting. For instance, here we have the 3 pentoses sitting close together, which makes a lot of sense. They go through the same metabolic pathway. So we get a lot of data out of that. And because, as I said, we're expanding that now. We do see that as the data set gets bigger, some of the correlations become a little bit more diffuse because, of course, not all fungi do exactly the same thing. There are these exception fungi, and they can really screw up your correlation analysis. So in some cases, we may have to curate by taking some of the really, let's say, strange fungi out of there to just make sure we keep seeing the big patterns. But we hopefully will publish all of that this year, and then you can basically have a look if you agree with it or not. The nice thing is that the database contains the original pictures, so if you don't agree with our assessment of growth, you just do your own. So this is basically why we decided to present it as such. Now I want to talk now more a bit about regulation, because for me that's actually the topic I'm most interested in, because I think these enzymes, they will tell you what the fungus can do, but regulation very much tells you why the fungus does it. Because regulation ensures that the utilization of the polysaccharides is done efficiently by producing the right type of enzymes. But of course, it's really important for the fungus that when it's degrading a polysaccharide to release a certain monosaccharide, that at the same point it's also able to consume this monosaccharide and to basically convert it through its metabolic pathways. So regulation is also very much the link between degradation and utilization. Now, if we just look at Aspergillus, we have quite a few regulators. So before we had genomes, we had MER, which was the starch regulator, and we had XNR, which is revolving xylem and cellulose. Then we got genomes, so we had better ways to screen for a lot of things, and we basically got a whole bunch of other ones. So all the ones in green are the ones which have been identified, not necessarily all characterized to the same extent. For instance, the CR1, which has been picked up in Neurospora, it's probably involved in cellulose as well in Aspergillus, but it hasn't actually been studied yet. I'm not 100% sure that CR2, which is the homologue of MEN-R, in Aspergillus oryzae, if it actually will have the same function in 2 fungi. I don't think there's been any comparison done there yet. And for CLBR, this has only been studied so far in Aspergillus ecoliatus, and it's not really been looked at whether it has a similar role in other fungi. But we have quite a large set of activators. There's a couple we don't have yet, but we know they're there. So there's a general pectin regulator. We know that based on expression studies of the genes, but we just haven't been able to find it yet. And we also know that there's an aromatic regulator responding to fluoric acid, but again, we haven't been able to find that. Now, if you take into account that the average Aspergillus genome contains somewhere between 600 and 800 transcription factors, and we've characterized, not just on this topic, but in total, around 30 to 35 regulators, it's not so surprising that it's a little bit of work to find. But we're still trying, so we're hoping still to find more. We found a few other ones as well. What's quite interesting is that a number of these regulators are actually specific for Aspergillus. So there seems to be an expansion of regulation in Aspergillus compared to most other Ascomyces, even in some cases compared to Penicillium. So that's something which is quite unusual. Now then, in addition to that, of course, we have a repressor, CREA, which is conserved throughout the fungal kingdom, so it's obviously quite important. I personally find it very interesting because CREA has mostly been studied under a high glucose condition, and is being considered to be a repressor when you have, for instance, 3% glucose. Now in nature, I don't think many fungi ever find themselves in the presence of 3% glucose. So in nature, most fungi would find themselves, I would say, under carbon-limiting conditions. The fact that CREA has remained conserved throughout the fungal kingdom, it must mean that there is a role for CREA under carbon-limiting conditions. So this is something which I don't think anybody's addressed yet, and I think which is particularly interesting to have a look at. Now, so it's nice you have all these different activators responding to different inducers, but it doesn't necessarily mean that they all have their own set of genes. So this is just a snapshot of some genes which I've just put together here in this. So here we have 4 different inducers, so XNR, ERA-R, and these 2 unknown ones. And you can see that in some cases there are genes which appear to be regulated mostly by a single regulator. There are also ones like here, the arabinoxylin arabinofuranohydrolase, the beta-galactosidase, alpha-glucuronidase, which are regulated by multiple regulators. And in most cases, you can explain this by the fact that these enzymes are needed for more than a single polysaccharide. For instance, beta-galactosidase, which is sitting here, you find beta-linked galactose on pectin, on galactomannan, on xyloglucan, on xylan. So it makes a lot of sense that the enzymes responsible for releasing that are regulated by multiple regulatory systems. The problem is it does make the analysis of the expression of these genes particularly difficult. Because in some cases, like for instance in Aspergillus, we found that if we disrupt XLNR, we get activation of RR on xylose, whereas we normally don't. And if we disrupt RR, we get activation of XLNR on xylose, which is not the case, or on arabinose, which is normally not the case. So these two regulators apparently partially compensate for each other. So making a single knockout will not actually tell you what's going on because you get a compensation effect. And if you then have a gene which is regulated by both those factors, you may not find any, let's say, net change in the expression of this gene. Now, as I said, Aspergillus, RR is only in Aspergillus, so you would not find that direct, let's say, interaction in other fungi, but there are probably other situations like that in other fungi. So it's quite difficult to really see how this regulation then works for individual genes. Now, just to also show you a little thing, this is very old. This is from 1999 already. At that point in time, Crea was considered a glucose repressor. And this was a thing which I always, as a young PhD student still at the time, I was very surprised by, because I read these papers and people would say, we see higher production of enzymes on xylane than on xylose. And then the next paper would say, as was shown before, we have higher production of enzymes on xylane than on xylose. And nobody ever gave any explanation. It was just repeated that it was seen before and it was seen before. And then we thought we should be able to figure out why this is the case. And the obvious thing to do was to look at Crea. Of course, as I said at the time, it was quite, let's say, unaccepted, because Crea was really considered to be a glucose repressor. But we thought that maybe Crea might have something to do with it. So I was quite happy with this paper. This is the only paper I ever managed to do the experiments in 2 weeks for. So this was probably the highest output versus input ratio I ever had. But we did a very simple experiment, and this has been repeated later in other fungi like Trichoderma and some other ones as well, and they've seen more or less the same effect. So what we have here is we have Mycelium of Aspergillus niger transferred to different concentrations of xylose, going from 0 to 100 millimolar. And what you see, if you then look at 4 xylolytic genes, that as soon as the expression goes up, if the concentration of xylose goes up, the expression goes down. In some cases very dramatically, like here for alpha-glucuronidase and ferroesterase. In others a little bit more gradual, like for the endoxylinase and the beta-xylosidase. We fortunately had, of course, Crea mutants at the time, so if you do the same experiment, but instead of a wild type you use a Crea mutant, you basically lose all effects. So what this shows is that xylose is not just an inducer of the xylolytic genes, it's also a repressor, depending on the concentration. Now, of course, this changes completely how you need to look at it, because it basically means that the system I showed you in the beginning, where you have co-induction of the metabolic pathway and the extracellular enzymes when you have a bit of xylose there, it also, of course, has a double lock mechanism, because as soon as the hydrolysis of the polysaccharide goes so efficiently that more xylose is produced than can be consumed, the xylose will actually start acting as a repressor rather than an inducer and will shut the system down until the concentration drops again. So it basically means that we have this very nice balanced out system where we have induction through xylose, but it's concentration dependent. So normally we activate XLNR, then XLNR will activate the target genes, and this will basically get the whole cascade going. However, if xylose concentration goes up, or we have an alternative carbon source, we get activation of Crea. And Crea not only inhibits the target genes, but it also inhibits the expression of the regulator itself. So Crea acts very much through a double lock mechanism, acting directly on the target genes, but also on the regulators, making sure that no energy is wasted on producing enzymes which are not needed. And I think this is probably the crucial, let's say, reason why Crea is still there in all the fungi, because Crea ultimately is the energy saver for fungi. So this is basically what we then have in Aspergillus. So we have this very complicated system, so that we have, for instance, if we start from XLNR, so xylose induces XLNR, this will then produce enzymes which release more xylose, creating a positive feedback loop, but it also will produce enzymes which liberate other inducers like galactonic acid, ferulic acid, arabinose, galactose, and glucose, each of which, again, have their own regulator they activating. So we get this cascade of enzyme production related to all these different monomers. But however, on top of that, we have Crea, which basically makes sure that as soon as the monomeric carbon source concentration is high enough, it just shuts down everything until the concentration drops again. So very nice and elegant, and I think, to be honest, from an evolutionary point of view, it makes a lot of sense. Now, if we then look at these regulators, I don't have all of them here, I just put a couple of them up here. So if you look at the presence of them, then we start to see that there's actually a very large variety between fungi. And this is going back to my remark in the beginning where I said that I don't think that for plant biomass degradation you can talk about model fungi because they just differ too much. So which one is going to be a model if on one hand we see that there's difference in regulation, and then I don't have a slide on that here, but we just recently finished a study where we compared the effect of an XNR mutant in 5 different fungi, and we find that the target genes are different, the activation is different, the substrates they are affecting is different. How can we then assume that if we compare one species to the other, this is actually going to be a one-on-one comparison? So we need to be, I think what we should do, we should appreciate the differences and try to learn from the differences to ultimately get a better idea how plant biomass degradation can be so efficient. So what we see, so some of them are present in most filamentous Ascomyces, like XLR, MER to a somewhat lesser extent. NUR is already a little bit more restricted. ERA-R, only Urtiales, so Aspergillus, Penicillium, Talaromyces. We have this regulator here, GAL-R, which is involved in galactose breakdown. It's only in Aspergillus nigelans. We don't find it in any of the other Aspergilli. And interestingly enough, in some of the other Aspergilli, we find GAL-X, which regulates directly the galactose pathway, but in nigilans, GAL-X regulates GAL-R, which regulates galactose pathway. And on top of that, GAL-X and GAL-R are next to each other on the genome, and they're not that homologous that we can assume it's a local gene duplication. So regulation is highly, highly varied, and we really have to take this into account when we start looking at this topic. Now, just to give you a few examples, so here, for instance, we have XLR and ERA-R. So XLR we find in nearly all filamentous Ascomyces, except for the human pathogens. But ERA-R we only find in this group of Aspergillus, Penicillia, Talaromyces, and it pretty much has the same structure as the XLR for that group. Now, I just want to give you a few snapshots on some new regulators we've recently identified to again show you a little bit about genome evolution and how this can affect the overall picture. So we recently identified RAM-R, which is a Rhamnose-related regulator. It regulates genes involved in rhamnogalactone degradation, which is part of pectin. It also regulates L-rhamnose catabolism, so we again see this link between degradation of the polymer and consumption of the monomer. We see it in all Aspergilli, and we also find homologs in a number of other fungi, so it seems to be quite a general regulator. And if we look at the phenotype, we can see that it has an effect on rhamnose growth, which we see here very clearly, but we don't see an effect on pectin because we also have this other pectin regulator, which is probably compensating just like ERA-R and XLR for each other. Now then, what you can see here, so this is a synteny plot of a number of Aspergillus genomes, and what you can see very nicely is RAM-R is here in all the Aspergilli, and this whole locus is quite conserved. There's a couple of little jumps here and there, but overall this is a very, very conserved region of the Aspergillus genomes, and we also find a very conserved regulation. If we then go to the next one, which is this GAL-X I mentioned, so again we identified it by expression profiling. It regulates mainly the oxidoreductive galactose pathway. As I said, it interacts with GAL-R in Aspergillus nigelans, and we only find it in some of the Aspergilli, not in all of them. But interestingly enough, we don't find it anywhere else outside the Aspergilli except for nectar hematococci. So possibly, maybe this is an example of horizontal gene transfer, where we basically got the regulator from Aspergillus going into nectar. That's something we haven't really gone into detail in yet. But what's very interesting, so here we now have, so before we had a regulator which is highly conserved, we see it in many fungi, and if you look at the locus is very conserved. Here we have a regulator which is not very conserved, and if we basically, well, this is the phenotype, so we see there's effect on galactose and galactitol, not so much on anything else. But what I really want to show you is this one. So this is again a synteny plot in a similar way with the Aspergilli which has it, and you can see that the locus is completely unconserved. And I think in some ways using synteny as a tool for studying, let's say, conservation of regulation may be a very strong tool. Because you can assume that if the locus is very conserved, the neighboring genes are still conserved, you can probably assume that the function is still going to be very similar. Whereas the more divergent the loci are, even in related fungi, the more likely it is that also the regulatory system has been modified. Now we're starting to do this now for some other loci as well, just to basically see if we can see a general trend in conservation and non-conservation of regulatory function based on genome organization. Now then also another thing which I just wanted to point out, so MER, as I mentioned before, it was always described as the starch regulator, and this has been a starch regulator for a couple of decades now. When we started looking more closely into MER in Aspergillus niger, what we actually found that it doesn't just regulate things like alpha-glucosidase and glucoamylase and alpha-amylase, but it actually also regulates beta-glucosidase, alpha-galactosidase, and beta-galactosidase, but not beta-xylosidase. So actually what we found, it seems to regulate any gene involved in the release of alpha or beta linked glucose or galactose. And then when we basically tested this using some growth profiling, where we have again here, we have the disruptant strain, we have the wild type, and we have the multi-copy strain, we can see indeed the phenotype here on maltose and on starch. But we also have a phenotype on maltobiose, on raffinose, a little bit on cellobiose, and very strongly here on lactose, and here also on malacetose. So we can actually see that the regulation of these genes actually reflects itself in a physiological state for this fungi. So we may also need to think a little bit about these regulators, that maybe the original role which we gave them when we first identified them is maybe not the complete picture of what they're actually doing with respect to fungal physiology. Now the other thing which we found there, which was quite interesting, of course, the inducer of MER has been discussed for many, many decades as well. It's been suggested to be either maltose or isomaltose or God knows what. We had a bit of a look in Aspergillus niger, and Oryza has been shown very clear to be isomaltose. It wasn't so much the case in niger. And what we found here, that if you look on glucose, you can actually see that in the multi-copy strain, but to some extent also here at later time points in the wild-type strain, we actually get induction of the MER-regulated genes. But again, then we come back to this system which I mentioned before with xylose, that glucose, of course, on one hand is a repressor, but it can be an inducer as long as the glucose concentration is low enough. And in many cases, when people use glucose, they use 1 or 3%, and this is basically just under repressing conditions. If you drop this to below 50 millimolar, you actually get inducing conditions for glucose as well. So again, also something I just wanted to point out here, that the regulation and the induction thereof is not necessarily as straightforward as it's sometimes believed to be based on lab experiments. Do I still have time for this? Okay, then okay. I just wanted to finish with some comparison in Aspergilli. So of course we do a lot with Aspergilli, and because the nice thing what we have in the Aspergillus is we have a very broad genus with quite a large taxonomic difference, but we have quite a lot of genomic tools. So what we decided to have a look at plant biomass degradation of different Aspergilli. So what we did is we annotated the Casey set of the first 8 genomes we had. We did some synteny and ortholog analysis, some growth profiling, and then we started looking in more detail at the enzyme activities. Now, just to point out a couple of things here. So basically this is the content of the genomes for the different polysaccharides. Some of them are a bit higher, this one's in yellow. The difference is not really that huge. So even though, as I said, there's quite a large taxonomic difference, we don't find a really massive difference, exception being here in Aspergillus clavatus, where a large number of the pectinases seem to have disappeared. But that's pretty much the only really, let's say, outstanding difference in all these genomes. Then if we do the clustering, using the genome numbers as let's say, a target for the clustering, what we see here, we get this relationship between the species, and it fits very nicely because Fischeri and Fumigatus are close together, and they're also taxonomically close. Flavus and Oryza are close together, they're also taxonomically close. So we can more or less conclude that the content of the genomes with respect to plant biomass degradation follows taxonomy and evolution. So no big surprises there. We did then some growth profiling, and I basically just want to show a little bit here. So here we have a couple of these crude carbon sources, wheat bran and sugar beet pulp. And what you can see that all these fungi grow quite happily on these 2 substrates. There's a small reduction here for clavatus, largely due to the fact that it has much less pectinases, and sugar beet pulp is quite rich in pectin. But overall, all these fungi seem to like these 2 carbon sources. So we then decided to use these 2 carbon sources to do some enzyme profiling for these fungi to see if they then also produce the same enzyme set when you grow them on these carbon sources. And then basically we got very quickly the surprises, because here you just see 4 of them, and you don't need to read what's underneath here. Just trust me, this is about 20 different enzyme activities which we've measured. Just have a global look at the profile, and what you can see is that, for instance, if you look here at Aspergillus flavus, we get a lot of the enzymes here on the left-hand side, not so much on the right, whereas in the case of clavatus, it's the opposite. For Fumigatus, we get a bit of both, and we get a different again here. The ones in black are the ones which are produced in sugar beet pulp, the ones on white on wheat bran. So what we can immediately see, just by looking at this, is that the enzyme set they produce for degrading these substrates, despite the fact that they're growing pretty much equally fast on it, they're completely different. So they have a similar genome content, but they produce different enzyme sets. So they have a different strategy. Now, We then did the same thing again. So we then took the enzyme activities and we did a hierarchical clustering again to see if what we would find then. I'm not sure if I have the boxes. No, I don't. Basically, what we then see is that taxonomy is gone. So we have fumigators sitting down here, whereas Fischeri is sitting up here, Flavus up here, Oryzae down here. So it looks like, despite the fact that based on genome content, these species are similar related to that taxonomy difference, when you then look at the actual system they're using for degrading plant biomass, they use something different. And this, we think, is largely due to regulation. And it actually goes one level beyond, but I don't have those pictures ready, so I didn't put it in. We had proteomics done on all these samples, and we even find that in those cases where we see similar, let's say, endoxylenase activity, the endoxylenase responsible in the two species are not orthologous endoxylanases. So they have the orthologs, but it doesn't necessarily mean that the same ortholog is produced in two different species. So this really shows that even in closely related species, which have a very similar genome content, which have the same set of regulators, their response to growing on these polymeric substrates is actually completely different. They have rewired their system in such a way that it fits exactly their needs. Now, we personally think that this is due to the fact that although they live in similar habitats, their precise habitats in nature are slightly different, and their systems are probably optimized for that exact niche. Of course, we don't put them in their habitat in the lab. We put them in our lab situation, but of course they cannot help but respond in a way which they've evolved to respond in nature, so they will respond slightly different. Now, and this basically gives us this really nice difference. And so just to sort of summarize, so I think what I hope to show you is that these strategies for plant biomass degradation, they're hugely different in fungi. And this makes it sometimes very difficult to do good comparisons, but on the other hand, I think also we should appreciate these differences because it gives us so many options to use different enzyme sets, different insights from different fungi to ultimately understand much better what's going on. Now, we find the differences at enzymatic level and genome content level, but we also very strongly find differences at regulatory level. And we've actually recently, as I said, we've done this study with XLNR, but we see that even the same regulator in different species doesn't actually do the same thing. For instance, in the case of Nectoporta oryzae, we can't even find an effect on xylose inhibition. We can only find an effect on xylose metabolism. So this is really quite important to keep that in mind. But I think also if we are looking at a more applied point of view, we should make use of this diversity in using applications, because nowadays, if you look at any enzyme cocktail which is used in industry, it's almost always an enzyme cocktail from a single organism, maybe supplemented with one or two heterologously expressed genes. But maybe what we should be starting to look at is look at more in detail what these different fungi actually degrade from the different biomass. Because a fungus in nature has no aim to fully degrade plant biomass. Why would it? What it wants to do is degrade enough that it can get its carbon source, it can propagate itself, and it can basically stay alive. So there's not a single fungus in the world, I'm sure, which is dedicated to fully degrading any biomass. But this is what we want in our applications. So maybe if we can identify the different strategies which take care of different parts of the plant biomass, in application we could combine these strategies to some kind of enzymatic super cocktail to really generate applications which will get close to what we ultimately want to reach. And with that, I just want to leave it there. I just want to acknowledge, of course, Bernard Henrissat and Pedro Coutinho from Marseille, who have really done an amazing job with building the Casey database. And I think anybody in the field will realize that without the Casey database, we'd be messing around rather than being quite quick about it. And of course, all the people in my group who are really responsible for the data here, which I just have the pleasure of presenting, but of course I very rarely do any of that myself. So thank you for your attention.
Yeah, thank you, Ronald, for this intriguing talk. So I think one really big take-home message is that we should rather expect differences than similarities when we look at even closely related filamentous fungal species in terms of carbon source utilization. I was wondering if, looking at the Aspergilli, they are really cosmopolitan fungi. Is there any knowledge about different niches? Have people really looked into details? Where are the differences?
That's actually the big problem. I mean, I wish they had. And I was a little bit, when I moved to CBS, 5 years ago now, this was really what I thought I was going to be able to do now. I'm going to CBS, the biggest culture collection. I'm going to just mine that database, find out exactly where all those species come from and all those isolates come from, and then use that data to relate it to that. Unfortunately, up to very recently, it was not obligated for people depositing strains to tell them where they came from. So a very large number of the isolates in collections have no origin, and if they have an origin, sometimes the origin is limited to Japan.
Okay.
But whether this is a contaminant on a bottle of sake or a soil isolate is in many cases not indicated. So I think there is for some species and some genera there are maybe that information, but across the board we just don't have the information, I would say. At least I have not been able to find it. Maybe John would know better, but I have not been able to find a good source of a really large data set of, let's say, biotope information of fungi, which it goes to the detail which you need.
Yeah.
Because it's not enough to know that it's from the Pacific Islands. You need to know if it's from a tree in the Pacific Islands or the soil or a contaminant of food. And I think nowadays with the newer deposits, all that information is being included. So we will get there, but I would say not yet.
Yeah, I think it's not an easy undertaking. So you probably would also need to know about the quantities. I mean, when you can isolate one Aspergillus species from one site, then it doesn't mean that there is.
The only thing I could say though, and this is maybe interesting in that content, is that we have done a comparison on Aspergillus niger species isolates from across the world to see if they have to some extent adapted to their local environment. And we really had isolates from everywhere, from South America, North America, Europe, Australia, Africa. And we actually found they were still so similar that if we just grew them on wheat bran and we took a culture filtrate sample, we ran it on a protein gel, we could identify the nigers from any other black Aspergillus just by a simple protein profile. And the reason we think that's the case is because Aspergillus disperses very easily. So it's probably never really in the same biotope for very long. So it's continuously challenged with variation. In contrast, if you look, for instance, at some of the basidiomycetes, which are much more localized, what we there see is that we do get large differences in isolates, even though they can still cross, so they are clearly still the same species, but we get very strong phenotypic differences. So there seems to be a relationship between dispersal and specialization.
Yeah, interesting. Any questions?
John?
I wonder more about looking at many isolates of a single species. Have you tried the hierarchical clustering where you look at the enzyme activity on a number of items?
Haven't done the clustering. As I said, we've done the protein profiling, and I've actually, to test if I wasn't just wanting to see something, but if I was really seeing something, I had a few students in my lab at the time from a very low-level technician school we have. So this is pretty much people who haven't gone anywhere near high school even, so their, let's say, biological background is about as low as you could get it. And I showed them the pictures without any names with them, and I hustled them up a little bit, and I asked them I gave them one which I said was an Aspergillus niger, and I told them to mark which ones they all thought were Aspergillus niger. And all 4 students were able to pick all the nigers from that. So I would say it's not a quantitative test, but I think the protein profile is fairly strong in that. So I'm sure that if we would have done the cluster analysis, we would have also found that. But as I said, for other species which are less easily dispersed, like basidiomycetes, but also, for instance, species like Podospora and serena, which again also doesn't disperse so easily, we get much more variation between isolates, especially if they are isolated from very different locations.
Can you say anything about how the protein profile changes over time?
Yes, I can. We've done that as well, obviously, for several species, and it does change a lot. And I think this is actually one of the big flaws in current, let's say, industrial biotechnology. I think people have now more or less at least started to realize that we will never have one super cocktail which will degrade all the biomass. I think that stage, I would say, we've passed. People still seem to think that if you just throw the fungus in a fermenter on the right inducer and you harvest your enzyme mixture at a certain point in time, this will be able to do what the fungus can do by itself. And I think this is where a big thinking flaw occurs. Because we've tested this indeed, we've grown fungi on biomass over time, and we see a very strong modification of the enzymes as the substrate gets degraded further and further. And this of course makes sense, because the fungus will first take the easy things and then it will start taking the less easy things. We've also done this by using ring plates, where we looked at the enzyme production in the periphery versus the enzyme production in the center of a colony, where again you have that range as well. So you do get a very strong modification of the enzymes which are being produced over time, due to the fact that the substrate is changing. We have also tried this by spiking the substrate. Actually, that data is not anywhere near publication, because the students who did it showed it clearly, but I don't like the data in its form enough for a publication. But we spiked it, and if you basically make sure that you don't that you continuously give it a new substrate, then you can keep the enzyme profile quite constant because it keeps attacking the same part of the substrate. But normally you would see there's a large change over time. So that would to me also suggest that if you want to do industrial biomass degradation, you need to start with a certain enzyme cocktail and then during the incubation spike it with different enzymes to basically mimic this modification which the fungus does. So that's going to be, of course, quite tricky.
So that must affect your comparisons somehow because your comparisons are a single time.
Yeah, that's true. I would say the comparison I showed you was from one time point. We've actually done 4. So we did look at it over a different time. We chose this time point because basically based on the enzyme profile, let's say with the appearance and disappearance of enzyme pack, they all seem to be in their more or less steady state. But yes, of course, if you start comparing more, let's say, diverse strains or isolates which are growing at different growth speeds, you will have to correct somehow for that. And that's going to be very tricky to know what correction you need to take. John?
I wonder where you find transcription factors in one species and not another. You try relaxing the criteria or recognizing the models.
And they're really not there. As a GAL-R is the weirdest. I mean, I haven't had time yet to look in versicolor and Sudawi now that we have those genomes to see if it's there. We should do that actually, but I just haven't had time to go back to that. But what we typically do is we will do 2 cutoffs, a strict one and a loose one. And if we don't find it with the strict cutoff, we will have another look at the loose one and then usually do some manual annotation to be certain there. And in most cases, if you don't find it with the strict one, it's just not there. And of course, there are many homologous ones, but based on if you look at synteny and if you look at actual homology outside the DNA binding region, you just don't find any homolog, except for Crea.
Like enzyme turnover, over time, are you seeing the proteases are being secreted to turn over the enzymes that aren't needed anymore?
True, happens for sure. I mean, we see that too. We see enzymes going down at some points where we have in some cases now correlated enzyme sort of proteomics to transcriptomics to see this. And what we then see if we do those correlations, that there are enzymes which just continuously increase in their amount because they're apparently very stable. And there's low expression, but it's enough to just accumulate the enzyme over time. And there's other enzymes where as soon as the expression turns off, the enzyme disappears. So this is not, I would say, a species phenomenon. This is a very much an individual enzyme phenomenon. And some of them appear to be very, very stable. Some appear to be not stable at all. And this can still also depend on the growth condition, which probably then links to which proteases are being produced under that growth condition. But we've actually done also a protease analysis on the same samples which I just showed for the Casey analysis, and we see similar patterns there again, is that also with respect to the production of proteases, it's very different which ones are produced between the different species. So, yeah, I mean, that's a whole other factor which we usually don't look at so much. We did it because we had a project where it was suitable to have a look at proteases. But of course, in most cases, when we study the, let's say, the production of enzymes, we don't really identify all the proteases which are there as well and try to see what their functionality may be in relation to the, let's say, activity of the protein. So it's certainly, well, I often say to my students that life is not that simple. This is, I think, a clear example because we can catalog the Casey response, but then we don't look at the proteases, we don't look at the organic acids which have been shown to be instrumental in the efficiency of the Casey genes on plant biomass. We don't look at a whole bunch of other factors. So we're basically scratching the surface at the moment still.
So can you say anything more, or do you know anything more about carbon metabolite expression other than Crea?
Well, yes and no. There is definitely carbon catabolite repression outside Crea. We've seen that actually. We have some very nice evidence where in strains where we've knocked out Crea, we still see carbon catabolite repression. There is of course Cre-B, which has been described in Aspergillus nigelans. There are also some specific repressors like ACE1, although ACE1 seems to be a repressor in Trichoderma, whereas in Aspergillus it's a completely different function, and in Magnaporta it's a completely different thing again. So I'm not so sure about that one. So there does seem to be other forms, and I haven't seen any studies yet, and this is, I think, a little bit the pity in a way in the typical, let's say, fungal research field. Crea has always been looked at very much as a phenomenon in itself, and it's usually been looked at high glucose concentrations to really have a massive effect. And I think even let's say, the alternative carbon repression is being looked at under the same conditions, and is usually being looked at very easily testable target genes like the phosphatases or things like that. Unfortunately, I got a grant which wasn't awarded, which I really wanted to link to try and understand what is Create doing under conditions where you grow on plant biomass, where you have carbon limitation. Because that's, as I said, that's I think what happens in nature. And if Create is still there in all of the function, it has to have a function in there. Then also linked to that, you could then also start looking at alternative carbon repression under those conditions. Yeah, but personally, I'm very, very reluctant to compare anything to Saccharomyces, because of course most of the study in Saccharomyces has been done in the strains which have been in the labs for a long, long time. And we have been doing some evolutionary tests on strains to see how quickly they lose abilities if you keep them in the thing. And also I've heard from some plant pathogen groups. The most extreme case I've heard was that a very strong plant pathogen lost its ability to be a pathogen completely after 4 months in PDA. And we've seen similar, we've used it to our advantage. We've been able, for instance, to produce a mutant from Aspergillus niger which degrades cellulose equally well as trichoderma. Whereas Aspergillus niger is always considered to be a non-cellulolytic fungus. Well, it took us, I think, 6 months to make a mutant which can degrade cellulose and grow. It grows better on cellulose than phenylketones. And so there is, I think, with those kind of model systems like Saccharomyces, which comes from, let's say, a very domesticated background, you have to be very, very careful, because also MIC1 is actually, if you look we've done the homology for all the transcription factors, looking at Create, and yes, MIC1 is sort of a homolog, but if we use our normal cutoffs, which will pick up Create homologs in any other fungus, including Candida, Zygosaccharomyces, and any other yeast you look at, you will not pick up MIC1. It will actually drop below the cutoff. So I do agree to you with a point, but I'm very careful about judging anything based on the Saccharomyces system, because of course it's a very focused species which is just basically focused on hexose consumption. And that would require a very different rewiring of its regulation, and it could be that the reason MIC1 is so different is because of that history.
If there is no more questions, let's thank Ronald and people.